gentlemen, welcome to PhilSoc. It gives me great pleasure to introduce the illustrious and possibly, debatably, the greatest biology teacher on earth, Dr. Sam Crawshaw.
Thank you for coming. I'm staggered. One of our greatest scientists, SMGS, Hamza Rafiq, came up to me today and said, I'm not coming to PhilSoc, I'm going to see Mockingjay. So, thank you for trudging across Manchester and raining down into the field of SMGS, experiencing our wonderful, majestic Memorial Hall. Simon Jones is our development director, and his sole job is to promote MGS to the rest of the world. And he thrust a patent for me in my hand this evening and said, plug MGS. And what he meant was, he's just found out that the person who invented ibuprofen, which I'm sure all the women in here take at least once a month, is called Dr. John Nicholson, and he went to MGS, and this is his patent. So apparently MGS does produce good scientists occasionally, even if the teachers are bad scientists. Which brings me onto the title of my talk. And my talk is not actually, I'm not doing a Ben Goldacre bad science talk, but I wrote this talk 4 years ago when Ben Goldacre was going to do a PhilSoc talk, and then he didn't tell us until his agent wouldn't release it because he was busy finishing Bad Farmer. So this is the second time I've been drafted in at the last minute to do a PhilSoc talk, and I'm beginning to get paranoid that I'm just a stand-in DJ. Anyway, what I want to tell you about, because you lot are all about to go, you're all about to fly the nest, you're about to escape this dump and go and be scientists and work in science in some form or another. And because I did that for a bit before I became a teacher, I picked up various survival tips along the way, and some of them are quite useful. And I think the other thing I want to share with you is a bit of self-knowledge, because lots of people become doctors because their parents are doctors, and that's why they're going to be a doctor, and they're usually fairly frank about it. But I think it's interesting to know science is difficult, it's less well paid. Science is more unstable, works on short-term contracts for a long time, and people really lay their life on the line to do it, and some doctors then go into research and experience the same instability of, are my experiments going to work, what's going to happen? And so you've got to know why you're doing it, and it took me a long time. At the time when I chose science, I chose to because I didn't really know what I was doing, I was just following my nose, and now I do know. And when I think back to the things that pointed me towards science, it's quite interesting, because when I was a child, I used to be in school plays all the time, and I had a pivotal meeting with this man, the man who makes Russia laugh, who was called Popov the Clown. He was the most famous clown in the world. He came to Manchester to do a clown workshop and teach people how to be clowns. And my uncle, who is this guy, he looks like he looks just like my dad, worked at another television at the time, so he invited me to this clown workshop. So that is me being hugged in my Clockings t-shirt by Popov the Clown, thinking he was a bit weird. But I was impressed by his clowning skills, and so after that I started really getting into drama, and so that is me holding a bowl, being on a twist in the Lancashire Evening Post after I started on a twist, and then I was a cowboy in Oklahoma, that's me being curly, and then I was a Thai slave in King and I, and so I was genuinely, I auditioned for the National Youth Theatre, I genuinely thought I was going to be an actor. I did drama GCSE with my best GCSE results, and science was a long way from me at that point. And then, as part of my English GCSE, I read Pride and Prejudice, and I didn't really enjoy it, and I massively insulted the teacher who was teaching to me by telling him that I really didn't respect Austen's contribution to the English literary canon. And she was very angry with me, and I wrote an essay, which I got very poor mark for, where I've been quite critical of the book. And this subjectivity that art subjects have, where one person thinks one thing about something and one person thinks something else drove me absolutely crazy. I was absolutely outraged by the fact that my opinion was not valued or not correct or the right answer. And that's what science gives you, isn't it? Science and maths gives you that certainty of whether you like the other person or not. 5 plus 5 does equal 10. You are bound by that similarity. So I kind of moved towards science then. And I was ready, I kind of changed my plans. And I think one of the things that all scientific careers have is that sense of flexibility. I'm going to have to change. And often what happens during medical careers, what happens during research careers is you think you're working on one thing, and then you have to really be flexible and switch paths if you're going to survive. So flexibility is a real key in science, and new information changing the paradigm that you're working on and having to change your ideas about how things, is the Earth flat or not. Sometimes we've all got to accommodate the new ideas, and that happens especially in the biological sciences, but also physics, more than you would think. So I went to college and I was doing my science A-levels, and I was working in a bookshop on Saturdays, and it was I used to love that bookshop because people would come in all day and they would go looking for this law textbook, looking for this economics textbook, looking for this computer science C++ textbook, and I would help people find books and talking about their books, and it was really useful for identifying what scientists are like. And they're so driven and they're so focused, and they're often dressed appallingly and they're often quite lonely looking, but they're really excited when their book arrives on the subject that they're researching. And I found that kind of oddness but passion at the same time really amusing, and I've never lost that joy of being around scientists who are quite obsessive really about the thing that they're researching. And I used to pick up science textbook and look at things that I then realised were protein structures and metabolic pathways, and just go, I'd really like to be able to decrypt this information and understand this. And you will have that experience too when you go to university. Textbooks are not completely defunct yet, and you will be forced to buy a big fat anatomy textbook or a big fat biochemistry textbook and use it sometimes. And it's like entering arcane knowledge. They're not like boring A-level textbooks. They're really quite extraordinary to try and translate yourself and understand. So I had a pivotal evening where I watched a crime film noir drama called Stormy Monday, which is set in Newcastle and involves lots of passionate, deep conversations on those industrial bridges. And I completely fell in love with the industrial north. So I went to university in Newcastle and started doing my biology degree. I did my first year, it was just general science, there were physiology modules and cell biology modules and other things. And did my year and it went fine. And during the summer after my first year, which is 3 months long, I thought at some point I've got to get a job. So I got a job at a microbiology lab run by what is now United Utilities. And it's in this big building here, which is somewhere near Warrington, and that analyses water from all over the northwest. So sometimes it's farms where there's been an outbreak of some kind of poisoning, and they think that something might be getting into the water there. Sometimes it's from taps, sometimes it's from swimming pools, sometimes it's from just sewage works, water, anything. People complaining that the water coming out of their taps brown. All that water goes to this same place, and the processing plant does the same thing, which gets some water and pours it through this filter here at the top, and this is the vacuum pump. So that pulls water through a very fine membrane with pores in the membrane that are probably half a micron in diameter. So a bacterial cell will not, water will go through it, a bacterial cell will not. So all the bacteria are trapped on this membrane, and then you get some sterile tweezers, pull the membrane out and you put it in a petri dish, a mini petri dish, a small one, not the size you used. And you can see that the membrane is pink because it's got a pH indicator in it, it's got some nutrient media underneath it. And if there's any bacteria trapped on that membrane, they will grow. And if those bacteria are able to ferment sugars like lactose, they'll go yellow when they produce lactic acid. And that means they're a coliform, like E. coli. And E. coli in this system acts as an indicator that there's coliforms in the water. It doesn't mean they're horrendously E. coli They're an indicator organism. It means, traditionally, that something has deposited a lot of coliforms in that water, and it might be sheep, it might be human waste, it doesn't really matter. They might not all be pathogenic, but traditionally we associate coliforms with something that has come out of the alimentary canal of a mammal. So famously in this film, Fast Food Nation, they get a coliform count and they realise that the burgers that they're selling have got shit in them because of the way they're slaughtering the animals. So coliforms are an indicator organism. It means the water is not clean enough, and there are minimum and maximum coliform counts that are allowed, for example, in drinking water, but also in swimming pools, also on Blackpool Beach. And so there's a lot of monitoring of water quality that goes on. And so after my second year, I did some more work in what was then the Public Health Laboratory Services, now called the HPA. And I carried on doing some of that work, but I also did a bit more work in hospital labs. And some of that work was still looking at E. coli in the way that I've described there. But I would also get different samples, and they would sometimes be samples from people who had gone away, come back with gastroenteritis, and they would be sending in samples from their GP surgery or from hospital to find out what was causing gastroenteritis for this person. And if they are EHEC or enterohemorrhagic E. coli, they will be causing really quite significant damage to the gut. And you can see a picture of the gut with all of the things that we know about, like villi and microvilli. And if you make EHEC or really pathogenic E. coli glow, you can see that they're not just kind of flowing through the gut, helping us digest food, they're sticky, they're adhering very tightly to the sides of the gut, and they're producing toxins, some of which go into our cells, and our cells start lysing. So if your food or your drinking water is contaminated with something like E. coli 0.57, then your poo will, over time, slowly get redder and redder as it fills up with blood. And so when I was working in PHLS in hospital labs, the samples that I would get like this were from really quite ill people. And the first day I got put on the enterics bench to see if there was nasty E. coli in fecal samples. I said, who normally does this? And they told me a really quite disturbing story about a guy called Tim, who had been at the bench and he was dipping something into these pots, and then he was going to spread some of it out on petri dishes and see what grew there. And as he'd been pulling his sampling apparatus out of the tube, it just caught on the end of the tube and flicked. And it just flicked some feces right into the back of his mouth. And he'd just been so aghast and so upset. And he said, what am I supposed to do now? And they just said, go to your GP. And he went to his GP, and his GP said, I'm giving you all the antibiotics I've got. Have a month off. This is deeply traumatic. Please don't sue the PHLS. And so I was doing his job, and it really was quite an ecosystem in there. There's all kinds of things growing in there. And I also had a kind of the interesting experience of seeing tissue samples and biopsies from people who were producing this stuff, and it was pretty unpleasant. So working in a hospital lab was a really interesting experience because I could get a bit closer to seeing how we determine what is causing infections. And if you can ever get some experience in hospital laboratories, you really do learn an extraordinary amount about how the science we're doing here at school is applied. But there were other things going on in those labs, and whilst I was there, I made a massive effort to learn about as many different things as possible. So one of the things that was going on, pregnancy tests that we could buy on the shelf in Tesco's now, 18 years ago, cost about £400. They were really expensive, and yet every woman in a hospital who's fertile or of childbearing age has to have a pregnancy test before they have an operation. They often don't even know it's being done, but they have to have a pregnancy test. It's a legal requirement. And so there are loads, hundreds of pregnancy tests in a hospital every day. And the way I was doing pregnancy tests was with this black piece of plastic, and I would drop a drop of a woman's urine into one of those rings, and then I would drop some antibodies that recognise the same antibodies that you use now in pregnancy tests. They recognise human chorionic gonadotrophin, and you get a hallucination. So you get that kind of pattern. This is a great positive result. A agglutination is going, and it's going all kind of snowy and plaggy, whereas this is a negative result, where it's just remained kind of clear and smooth and a bit opaque. So I was doing loads of pregnancy tests, and then I would literally ring up operating theatres and go, she's not pregnant, you can operate. And it was really funny, because just occasionally I would ring up and I'd get a funny sample, and it would have the paper wasn't quite right, but I would ring up and go, yeah, I got this sample, she's not pregnant, and I would hear a nurse at the other end, and I go, oh, thank God. And it was just really funny how there was this kind of black market team in nurses saving huge amounts of money doing pregnancy tests through the system. Anyway, apart from doing pregnancy tests, I spent a few days working in a hospital as a porter, and that was perhaps the most illuminating bit of all, because sometimes there are often porters who are specially assigned as runners in hospitals for really high value samples. They don't do anything else, they just take that sample and run through the hospital building and get it to the right desk as quickly as possible. And so often I would get blood samples from someone who was HIV positive or had tuberculosis, and they were in a really bad way, and we need a very quick diagnosis. And some of that would be done through microbiology or microscopy. Sometimes you would get tubes like that and just put them in an incubator at 37 degrees, and one would be aerobic and one would be anaerobic, and they would just rock gently. And if the patient's got septicemia, if they've got bacteria growing in their blood, those tubes blood's the ultimate growth medium, isn't it? It's just full of amino acids and salts and sugars and everything we need. And so bacteria would grow very rapidly in those blood samples, and in fact the blood changes colour very quickly, and you can tell something nasty is growing in there. And of course there are urines as well. We've probably all given a urine sample at some point or another. It's just the easiest accessible fluid you can get out of a person, perhaps apart from saliva. So lots of urinary tract infections and lots of microbiology there, plating out urine on different plates and looking at things like this, which some of you will have done at school, someone's got an antibiotic resistant infection. We don't even know what it is. In a way, we don't care. We just try different antibiotics. And if we get an antibiotic that will successfully inhibit growth, we just say, don't bother finding out what it is. Just give them this antibiotic and this antibiotic, and their urinary tract infection will clean up, and they'll be happy. So that was another really interesting thing. Seeing the lung of someone who smokes is fascinating, because it really is just a totally different colour. It really is full of tar. So I love seeing stuff like that. And on Thursday, the Chorley Seamans would arrive, and they were from infertile couples who couldn't get pregnant. And one of my favourite jobs, just extraordinary pleasure, because it was so life-affirming, was to take a swab of someone's cervical mucus and dab it in the middle of the microscope slide, and then get some semen from their unhappy husband who, they weren't able to get pregnant, warm up his semen in an incubator so they all started swimming again, and then dab rapidly swimming sperm around the outside of this cervical mucus. And if those two people are compatible, if sperm are going to be able to burrow through that woman's cervical mucus, what you get are these amazing patterns. This is mucus. These are obviously sperm cells, and they're burrowing, they're making huge channels through the mucus, which is exactly what has to happen for conception to occur. And so sometimes you would get that pattern. Sometimes if you left cervical mucus surrounded by sperm cells for 2 hours at 37 degrees, all the sperm would still be swimming very happily, but they just wouldn't be burrowing through the mucus at all. And those couples are not going to get pregnant unless they have some kind of treatment or sperm are deposited inside the uterus, and that can often solve that problem. So ringing through those results as well was a real pleasure. And I also did some work at night times, because some of the sampling that's done for hospitals like that has to be done at night. Sometimes patient stuff is done at night, but also if you're doing tidal work, checking whether Blackpool Beach is safe to swim in, the tide's only in at certain times of day and night, so some samples come in at night and have to be sampled at that time. So I can still remember all of those things really vividly. I didn't really care about the money, I did get paid badly, but it was all about the experience, and seeing people in those labs and what they were earning and how hard they were working was really valuable for my career. So I finished my degree, and I was still living in Newcastle, and all of my housemates, all without exception, moved to London and got really boring jobs. And they kept coming back from these milk round days going, yeah, I've been snapped up by Deloitte, and I'm going to earn loads of money. And I was just, the thought just filled me with horror of helping people avoid tax for the rest of their lives. I just couldn't. Just kept going to non-crowd places and ended up in the pub. I just couldn't do it. So in the end, I signed up for a PhD, and that was mostly because during the science practicals I've done, the people who were demonstrating, the people who were helping us with PhD students, and I just found them fantastically interesting and really passionate about science, and I really wanted to spend more time with them. So doing a PhD was largely a process of avoiding getting a job and wanting to spend more time with people like that. That's really why I did it. And I applied for PhDs in lots of different parts of the country. I went to see all those people, and in the end, stayed in Newcastle. And it was partly because Newcastle was just mad at the time. Kevin Keegan was managing Newcastle United. That's Sir John Hall, who was trying to build the Geordie nation. He wanted Newcastle to be different and only have players from Northumberland in it. And they'd just signed Alan Shearer for the highest Premiership transfer fee of £15 million. And I loved Newcastle, so it was partly the place that made me want to stay, and it was partly the project, which I'll tell you a bit more about in a minute. I did a dissertation in a microbiology lab, in a particular lab that was working on activated sludge. And activated sludge caused a really big problem in lots of parts of the country where sewage reaches activated sludge plants, and we aerate it. That's what causes decomposition, isn't it? When there's lots of oxygen, decomposers can respire and do their job. So these things here are propellers. They're just whizzing round and aerating this dirty, brown, murky water and allowing decomposition to occur, and then it all goes into a river. That's what a sewage treatment plant is. But on some sewage treatment plants in some parts of the country, not everywhere, they get this floating mat that looks like a kind of chocolate brown floating on the top, and it doesn't really do any harm, except when it's really windy, sometimes the foam will blow off and land in children's playgrounds and primary schools nearby, and that's a bit of a public health risk. So I did spend some time in my third year doing my dissertation. That's me, visiting a sewage treatment plant, and that's Fiona, the PhD student who supervised me, getting samples and then taking them back to the lab and looking at the organisms that are in there and trying to break things up to establish what's in there. And I was really into microbiology, applied microbiology, so I was learning, for example, that we can use very simple mechanical, the end solution for this stuff wasn't microbiology, they just used to scoop the foam off and burn it. But I was learning that practical applications are interesting and satisfying, and I was kind of seduced by the person who ran that lab, that there was a bigger problem that I was going to help solve, and it came actually from Eastern Europe. It came from places like Chernobyl, where there had been big nuclear accidents, and there were lots of radioisotopes in the soil, radioactive heavy metals that were permanently polluting the soil and were very difficult to remove in large areas. The Sellafield is the same Sellafield has not had a nuclear accident, but if you point a Geiger counter at the ground, it's really radioactive. Sellafield had to spend millions of pounds a year preparing for when Sellafield closes, which it inevitably will, and what they're going to do with all the nuclear material that's still sitting in a car park outside the nuclear facility, and also how they're going to clean the ground and people are going to be allowed to walk on it. And so I was basically a beneficiary of some of that money, in terms of, because being a Welsh have to spend millions and millions of pennies to them. And I used some of that money to look at strategies for cleaning up soil. And we were copying the Eastern European very cheap solution, which is to grow sunflowers in contaminated soil. Let the sunflowers soak up radioactive heavy metals and then burn them. That's the Eastern European solution, which is, again, cheap and effective. But we were looking at, well, how can you optimise that process? What could you put in the soil? Are there microorganisms that would secrete certain acids, that would solubilise certain metals, that would speed this process up or not? So the idea of my PhD was partly to do that, but it was also, because this was 1997, it was also the era in which genome sequencing was really kicking off, and the first bacterial genomes were just arriving, and all genome projects came up with the same problem. They would all come up with data like this, where they would analyse the genome and they would get 4,500 genes, something like that. And a third of the genes they would know about, they would know that, that's a DNA polymerase, that's an alpha amylase. And then another third of the genes they could easily allocate and say, well, I don't really know, but I've analysed the sequence and it's just the same as an amylase, so it's probably just another slightly different alpha amylase. And then they would also have a population of genes, over 1,000 genes, and none of them would have a clue what they did. And we were always saying, we were always looking at these organisms and going, well, we're talking about organisms that people have been studying for decades in labs at 37 degrees on Petri dishes. And if we want to know what all these genes do, we need to put these organisms back in the environments where they're naturally found, which for this organism, for Bacillus subtilis, was in soil. So my project was saying, let's see if we can track, let's see if we can follow gene expression in soil. Which genes are expressed in soil? Are they important and do they enable organisms to survive? If we knock them out, if we mutate them, can that particular strain that we've made still survive in soil? And does it associate with plants and things like that? So what I learned on my PhD was the skills to make genetic cassette files. I would stick loads and loads of genes together. That's GFP, some of you know about that glows in the dark. That's a chloramphenicol resistance gene, chloramphenicol acetyltransferase. Xylene is something that if you grow colonies of bacteria and spray them, it makes them glow yellow. So I can say, oh, that's one of mine, I've got that gene in there. And then this thing, I started making barcodes as well, molecular barcodes. I just made up some sequence that didn't appear anywhere else and said, I'll be able to find it now. I can put it in the soil and I'll be able to find it again later. It's like barcoding your kids and then sending them off into the wacky web to play for a couple of hours. So I was busy making strains of bacteria, sticking them into the environment, just letting them acclimatise and compete with each other, and then seeing which ones would survive and see which genes were being expressed. And it was really interesting, and I learned lots of kind of interesting things about some of those genes of unknown function. But for me, the real importance of my PhD was that I gained skills. I gained molecular biology skills about cutting up and sticking together different bits of DNA. So, and you know, the other thing I've been doing during my PhD was actively reproducing and having lots of genetic experiments of my own. So, by the time I finished my PhD, I had a child and a half, and the second one was going to be born soon. And I said to my wife, I finished my PhD, shall we move to America? And I'll have a really great career and become a professor and earn loads of money. And she said, well, that's great for you, Sam, but I won't be able to get a work permit and I've just had a baby, so I'll be at home with a baby and a half all day whilst you are working 14-hour days in the lab. And I had to concede it was a fair point. So instead of moving to California or Japan or Germany, which is what lots of scientists do at that point in their career, we moved to Manchester. And what I was doing at this point was thinking, I was still thinking about my career even though I wasn't going to America, and I was thinking, well, I've got, you know, 8 papers for my PhD, but for a good scientific career, what I need are lots and lots of high-impact factor papers in top journals that are peer-reviewed and respected internationally. So when I was selecting projects at this point, I wasn't even going, what am I interested in? I was going, I want a well-funded lab run by a really organised person who is powering out papers at high speed. If I'd stayed in that field, if I'd stayed doing genomics, the problem with genomics is to analyse all the genes being expressed all at the same time. It's such a huge enterprise. Your papers usually have about 50 people, and they have bioinformatics people in Oxford and Cambridge, and they've got people that are providing you with machinery, they have people from BFL, and it's not very good for building your own scientific career where you are the key person enabling discoveries to happen. So I moved to Manchester, I got a job in this building, which was being built when I arrived and was finished in 2004. That is the Michael Smith building. And if you drive past it now, on the A34, there's a big tube sticking out of the side here going into the brown Stockford building, so they can move rabbits about without animal rights activists harassing them. So, but that building's really nice. I mean, you know, Manchester University had just merged with UMIS, so it was temporarily the biggest university in the country, University of the Year. There's loads of money sloshing out, so it was a really good time to work. And it makes for an interesting comparison talking about budgets and money, because Mr. Freeman, who was another biology teacher here, who I love dearly, who we haven't even met at this point, was working in Mauritius, and he was doing conservation work, and doing some really, really interesting stuff, which I'm sure he would give you a PhilSoc talk about. But what's so funny is, because it wasn't going to solve the problems of a multinational industry like B&FL, or because it wasn't going to solve a medical issue that rich people will pay money to solve, his funding was very minimal, and actually he was kind of living almost homeless in this shack with several other people for 2 years. But as you may know, if you know, he loves climbing. He basically climbed up trees all day and tagged birds and did all kinds of things. So you can say to Mr. Freeman on Monday, I saw a photo of you topless on Friday, and just see if he knows what you're talking about. Anyway, so I moved to Manchester, and I got a job in a different lab as a eukaryotic cell biologist. So I was working on animal cells or human cells. And the irony is, I knew absolutely sod all about it. I had done animal biology like you, I'd forgotten all of it. I've been working with bacteria all of my career so far. So what's interesting is, the guy running the lab said, I don't care about any of that, I just need someone who can stick bits of DNA together. I've got loads of cell biologists, but they're all crap at molecular biology. So as soon as I moved into that lab, I solved all kinds of problems, and I finished off lots of papers. I got my name on loads of papers really quickly. It was exactly what I wanted. And so that's a really useful lesson in science, is that it's not knowledge you have, it's skills to solve problems for other people. And the skills now that I think are sexy, I think lots of companies like AstraZeneca don't particularly want cell biologists. They want in vivo biologists, people who can work with animals for phase 1 clinical trials, where they're putting stuff into animals and seeing if they die or go drink or do something else. And lots of medicine things in the first book is neurology based, and that often involves doing stuff with primates as well. I think there'll always be work for computational biologists, and there's not many. Biologists generally don't like maths and don't want to do things with computers. So if you can cross that boundary, then that's really, really useful. And of course, chemists never go out of fashion in the pharmaceutical industry, because ultimately your job is to make chemicals. So, I moved to Manchester, I was working in a lab, I was doing research, and I found this old photo of just 3 other people who were all in the lab at the same time as me. The first one, Helen, did about a year of postdocing and then became a medical writer. And I think he's now still working in Cheshire in one of those kind of satellite companies around AstraZeneca. They basically write up data for Zeneca. John was doing a PhD, he then joined the MOD. He now works for a bank as a kind of analyst about securities and stability in the Middle East and things like that. The last one, as I heard someone whispering, moved into teaching and has just left Whitney as was the head of chemistry for a few years, and has now moved to London and has baby. That's Claire Spinks, I think she's called, she was Duggan. So there are lots of offshoots to research, and lots of people do a PhD and then bail out. Lots of people do a couple of postdocs, like I did, for 3 to 7 years, and then bail out. So it's skills that matter, not knowledge. Memorising lots of different parts of the cell is not really that useful or impressive. And the second thing that I learned from being a postdoc is whenever I did an experiment, and they were all very interesting experiments, I'd do it once and then be bored, and I'd go and see my prof and say, this is what I've discovered, it's really great, let's publish it. And he would just send me away and say, you're going to have to do it again and again and again, 6, 7 times before I believe you. So actually, a lot of postdoctoral work, thinking about it was really interesting, but actually the people who think about it are the profs in the labs who are writing grants. The grunts in the lab are just grinding out research data, and it's quite hard work, and it's not always particularly exciting, but I'll just try, I've got about 10 minutes left, and I'll just try and give you a flavour of what I was doing in that lab. This kind of cell, this model cell, was horrendously complicated to someone who was used to working with bacteria. And the kinds of problems that eukaryotic cell biologists are often trying to study, and the things that cause disease in eukaryotic cells are often a protein is synthesized on the ribosome. The ribosome might be in the cytoplasm, it might be on the rough endoplasmic reticulum, but it's unlikely that that protein is just going to end up in that space. The protein's got a specific destination where it's supposed to be: the lysosome, the Golgi, the nucleus. the cell membrane, the cytoplasm. We don't know, but the cell does know, and the cell knows because of a specific signalling mechanism which this guy Logan discovered, and he also got a Nobel Prize in 1999 for the signal hypothesis. Cells have, proteins have specific sequences within them. They might be the end terms, they might be the beginning of the protein, they might be the end. But those specific sequences say this protein should be in this bit of the cell. It's just like giving certain age groups at school certain ties or certain uniforms. It enables teachers, it enables the cell sorting machinery to know where things should be. So in my lab, they were looking at the idea of protein being synthesized. This kind of two blobby, I made this diagram by the way, and I spent hours on this very subtle shading. I can't tell you how to paint that. But this is a ribosome, and this is a protein coming out to being synthesized. So we can't see the messenger RNA, but it would be there coding for this protein. And this protein's got a signal on it that's being read by this thing, which is signal recognition particle. And this might be saying, oh, it can stay in the cytoplasm. It might be saying it's going to be secreted, so it needs to cross this membrane, which is the endoplasmic reticulum membrane, and go to the movement of the endoplasmic reticulum, and then go off in a vesicle, and go to the Golgi body, or go to the lysosome, or go to the vacuole, or go somewhere else. So those signals are read, and if it's going to be trafficked, if it's going to be transported somewhere, it'll have to go into the membrane, or it may go through this structure, go through the transluminal, and rather than going into the lumen and being secreted, it may actually exit sideways. It may go into the membrane, because it might be a membrane protein, like a hormone receptor or something for active transport or for facilitated diffusion, all these concepts we've learned about. So there are lots of different destinations, and things go wrong in cells. Diseases happen when proteins don't reach the destination they're supposed to get. Or diseases can happen when proteins are made, but they don't fold correctly. They end up the wrong shape, and then they've got to be degraded and got rid of. We can't just have the cell full of crap hanging around, and we've got to get rid of it and degrade it. So that is 7 years of research, 7 years of my life, and I'm just going to give you an example of the kinds of experiments that you would do to look at that stuff. So this is a ribosome again. This is a transluminal, and it's making a protein that is a multi-spanning membrane protein. You can see there are lots of transmembrane regions threading in and out of the cell membrane, and that's what receptors that drugs bind to. That's what receptors that bind to hormones, that bind to neurotransmitters. That's what they look at. They're big and they're complicated, so that they can recognise lots of different shapes. And what we wanted to know was, one of the things we were looking at was, when this is threaded into the transluminal, how does it get out of that transluminal into the membrane? Because that's a really energetically complicated thing to do to cross hydrophobic and hydrophilic structures. So we would be getting, this is the crystal structure, you can see alpha helixes here. This is what the transluminal looks like. The ribosome would be sitting on the top here, and it would be pushing a new protein right down the middle. And what we felt was, there's actually three different polypeptide chains there. There's one there called beta, there's one there called gamma, which goes like this, and then the other one is big, alpha. There's lots of transmembrane domains. But we felt that we could see a way that a newly synthesized protein would go into here, and if it was going to be secreted, it would go right through to the lumen and go to the vesicle and be trafficked. If it was going to be a membrane protein, at some point it would have to push outwards into the membrane. And we couldn't work out if something had 7 transmembrane domains, do all 7 go into here, and then do they all go together? Or do they pop out one at a time? And that was a molecular question that we thought was an answerable question. So we wanted to follow the movement of a newly synthesized protein into the membrane. We were using this protein opsin, which is found in our eyes. It's a photoreceptor. It's usually bound to a pigment. And this is, you can see these are lots of different amino acids. These are alpha helixes and in different transmembrane rings. And I spent a lot of time mutating this so that I could knock out lots and lots of cysteine residues and turn them into something else. So that when I was finished, there was only a single cysteine residue here, which which I could use as a kind of probe. There's some glycans there, there's some lipids there, pancreatids, and the bio as well. And one of the things I did with my molecular biology skills was mutate the whole thing as well, remove 7 of the transmembrane bits and just put some soluble stuff on the end instead. And I was comparing what happened during the integration process if there was 7 or if there was just 1. And so I would synthesize proteins and radiolabel them, and then add some glue and see what they were next. So in this protein, where there are lots of transmembrane regions, that's the newly synthesized protein being made. If I added this glue, it would stick to things like, this is the transmembrane component alpha, this is the transmembrane component beta, and it arrives in the transmembrane and sticks to these products for a bit, and then eventually it loses them and disappears, and that's probably because that first transmembrane is being pushed out into the membrane further and further away from the transmembrane. Whereas if I used my own weird mutated version, that didn't have any subsequent transmembrane domains, then it stayed, it kind of hung around the translocon for longer because there was nothing to push it out into the membrane. We felt that was like saying there is a sequential model of integration where one transmembrane domain goes in and then it gets pushed out by the next one, which gets pushed out by the next one. And we just built and built on the data set and made models for other people and ourselves to test. And we also looked at what happened if we synthesized the protein, but it was crap, it was misfolded, it fell off the ribosome and ended halfway through a transmembrane region. We would synthesize it and then let it go into a membrane and see what that stuck to. And that stuck to all kinds of different things. And we postulated that was the cell's cleanup machinery that was getting rid of this crap that's not proper protein. And so I would then have to email lots of labs and read lots of papers and find out which antibodies were available to components in the endoplasmids particularly, and see if I could identify any of those structures. And eventually, after lots and lots of messing around, I was able to identify some of these products that appear when you release crappy half fragments into the membrane that are obviously going to be degraded. And so I was able to work on those in more detail and find inhibitors to them. That's really key to disease and disease pathways. So, all the time I'm thinking, paper, I've got to sustain my career, I've got to get some more funding. And I'm always thinking, if I don't find out what any of these things are, I'm not going to be able to publish, because I can't publish a paper that says, there are these things, but I don't know what any of them are. You have to have positive data to get a publication. And that's a great pressure for scientists, and it also leads us to kind of bend statistics, because we're always desperate to show that something is very significant. And I felt that pressure all of that time. But of course, I still loved it and carried on for quite a long time. So that's a real lesson about the dangers and the pressure of science. But all the time I was really enjoying it, and I was turning up working genes. I didn't have to wear a tie, didn't matter what time I turned up. If my wife phoned me and said, I'm about to throw your son down the stairs, I could just say, it's all right, darling, and put my stuff down and go home, and I could do it on a Sunday. So that flexibility and that kind of following your nose and just seeing what happened was something that I really enjoyed about science. In the end, the reason I went into teaching was because I reached the same point in that lab where I've been working there for 7 years, and to carry on, I would be setting up essentially a competition with my own boss. And so really I had to go somewhere else. And at that point I had even more kids and they'd all started school. And I realised it was cheaper to retrain and become a teacher than it was to, for example, move to the States or go move to another lab to get a lectureship. So there are challenges associated with research science, and it is unstable, but it's also just really different and really rewarding. The people you meet in these labs remain as interesting as they ever have been. So I will stop because it's 1 minute past 5, but I think I was looking for kind of motivational science quotes, and I really like this one, from this person's favourite scientist that we're all supposed to idolise, and I do like science, I know one does. But what's so interesting is his statement that most of the time he was wrong, most of the time things go wrong in science, and you must enter in that spirit that I'm prepared to be embarrassed and depressed quite a lot of the time. But things do go well too, and they're really enjoyable. So I hope that's been a kind of interesting insight into research. I really encourage you to do PhDs. You'll be working for the rest of your life anyway. So if you can do a PhD and expand your student experience for a few more years, it's well worth it. Thank you very much for turning up on Friday. I will see some of you on Monday. The rest of you I will see on another Wednesday. Thank you very much. Well, you can ask me questions if you want. I bet you all want to go see Monica and JB. Does anyone have any questions? I won't repeat it. Go on, Vlad. Oh, yeah. Well, it's only that the labs that you work in are better. Because the thing about ecology is it's really cheap to fund as well. So there are plenty of ecologists, there's plenty of conservation studies, but it doesn't yield really well-paid jobs. That's the problem. And in terms of research, the problem is, if you work for a pharmaceutical company, you do always end up researching problems like depression or sleep apnea or diabetes, which are all basically rich people's diseases that don't exist in poor countries. And you're far less likely to be researching. I mean, there's not a lot, there's not a lot going on for tuberculosis, malaria, HIV. If you look at big pharma, that's not what's going on at all. There's lots of cancer research, and cancer is a very great disease to solve. But they're just. I know when I'm out of my depth, and when I'm talking about ethics, I'm well out of my depth, especially if Max Rogan's here. But I think that there's a discrepancy, and you can't really do anything about it. There just is. Yeah. Go on, Josie. No, it's not possible to cure HIV because it will always mutate faster than anything. Lots of great vaccines have been made to HIV, and they're just outdated and useless within a year. If you really wanted to solve HIV, you could make everyone in the world only have one sexual partner in their life, in which case HIV and a million other sexually transmitted diseases would disappear within a generation, completely. So there are very easy solutions, it's just they're not very realistic. So, no, sorry. Go on, Max. Can HIV be passed on to a kid? No, but the worst case scenario would be that child would live long enough to be of reproductive age and pass it on to one person. So it would still be Max. Yes. Yeah, I know. It's not a brilliant solution, but it did clear up the soil a bit. You still got radioactive heavy metals, that's always the problem. MGS boys just help you with the floor, as always. Go on. Yeah. Yeah, no, I just don't agree. I just think having a job, whatever your job is really boring because you have to work really hard. And a PhD is a kind of great in-between house, isn't it? And you'll see what I mean when you get to university. PhD students are the most interesting people around because they've got lots of free time to think and to follow their interests. And you can't help just go, and they're all really clever. So you can't help just go, I'd much rather do that than be sweating away at the age of 22 to make money for someone else, So it's you only have one life, and yes, you're right. If you really want to pursue money, then it's probably not necessarily. Although I get paid more at MGS as a teacher because I've got a PhD. So PhD students do get PhDs get paid more money in the same way that people with first class degrees get paid more money. So, but yeah, you have to do it for the sake of doing it, not necessarily as a progression to something else. It's just no, not at all. No, because I was just up for it. I just loved messing around in the lab and doing stuff. And actually, there were other PhDs I looked at that were funded by industry that had a much closer commercial interest in the outcome. And those PhDs were usually a process, a fermentation process, something, and they were just tweaking one variable at a time, trying to grind 1% more efficiency out of the process. And those people then got jobs very quickly. And I'm sure they were well paid, but their PhDs were so boring. You know, they were just really boring. Whereas my PhD, where I'd just go and go, I think I'm going to have to go on the microscope again. I'm going to put the microscope. And I never felt more like a scientist than when I was going to have a go on that machine. And that's what science should be like. So a good PhD should not bore you at inception. You should see a bit bigger. That's such an interesting problem. And this lab is full of such interesting people. I could spend all day talking to these people, and then that's what you're doing. It's the only job where sitting around drinking coffee, kicking ideas about is a really valued bit of the process, because that's how scientists work, isn't it? Through collaboration and sharing ideas, go, oh, I could do that for you, and then you thought, what have you seen the paper, you thought about this? So it suited me down to the ground, you know. I have absolutely no regrets about doing a PhD. Don't have boring office workers put you on. Right, I'm going to shut you up, Joseph. It's one of my only opportunities. Go and enjoy your weekend. Thank you for coming.